As the bacillus is going into the lung, it will enter the alveolus, and in the alveolus we can see macrophages, and they simply uptake this bacillus within them to process it. So we can call it a antigen presenting cells. Now look at the picture again, back into the PowerPoint. This macrophage will process this antigen, the tuberculous antigen, with the help of class 2 antigen of MHC. Now this combination, they will activate the T cell receptor of a thymocyte or CD4 cell. Now once the CD4 cell is activated, they produce various kind of cytokines, and the most important cytokine in this particular scenario is gamma interferon. Now what is the function of this gamma interferon? This gamma interferon will modify the macrophage to produce epithelioid cells. Did you get it? This gamma interferon will modify the macrophage to produce a activated macrophage, which is known as epithelioid cell. Suppose you don't want to use the word epithelioid cell, what is the term you can use instead of that? We can use the term activated macrophage. Now this activated macrophage, why we are calling it activated macrophage? Because this macrophage contains tuberculous bacillus within them, and they destroy them. How they destroy them? They destroy them with the help of nitric oxide, NO. So let me come back to the previous thing which I wanted to discuss. So which particular chemical mediator is responsible for the formation of the epithelioid granuloma? It is gamma interferon. So this gamma interferon will modify almost all macrophages which are available in that area. Not only that, it will pull some macrophages or monocyte from the blood to produce macrophage, and then they modify them, and ultimately you get aggregates of this activated modified macrophages, and this aggregate is known as granuloma. Now what about the caseation necrosis? As I told you, this epithelioid cell is a cell in which you can see plenty of tuberculous bacillus. Now this tuberculous bacillus will be killed by this epithelioid cell by nitric oxide. So there is a kind of fighting going on inside each epithelioid cell, and as the fighting going on, both epithelioid cell and the tuberculous bacilli, both of them will be dead. They will die. Both of them will die. And as they die, you get the dead products, and this is known as caseation necrosis. So caseation necrosis is a product of death of both bacillus as well as the macrophage. So what are the points to remember here? Mediator that is responsible to produce a granuloma is gamma interferon, and how this macrophage destroy or kill this bacteria, what is the mediator for that? That is a nitric oxide, and these are the two most important things you must remember from this pathogenesis. So here once again a simplified diagram. APC cell present the macrophage, present the tuberculous bacillus, and now this will activate the CD4 cells. Now activated CD4 cell will produce gamma interferon. They will modify the macrophage to produce epithelioid cells, and when many epithelioid cells join together to produce aggregate which is known as granuloma, within the epithelioid cells this macrophage produce nitric oxide, and this nitric oxide not only kill the bacteria but also will destroy the macrophage. So the dead products of this macrophage and the bacillus, they produce caseation necrosis. So at the center of each granuloma we can see the caseation necrosis. So have a look at this particular picture. You must identify the picture. You can see a nice giant cell here. There are some epithelioid cells over here, and if you look at this particular place, this place is amorphous, granular. They are containing dead tissue, and this is the area known as, yes, what is the name of the area should be known as? This is how the caseation necrosis look like. And if we see the caseation necrosis, what is the possible diagnosis of disease that we see here? We call it a tuberculosis. So up to this much today. After that we'll come back and discuss the rest of tuberculosis. We will show you how the granuloma look like. This is what we saw here is a microscopic appearance of a granuloma. We want to see how it look like on a gross appearance. We'll come back and discuss the secondary tuberculosis as well. Thank you.